blood cell. Erythrocyte, erythro means red, cyte is cell, so it's the red blood cell. That number is important for you to know, 4.2 to 6.2 million. The exact number is not as important as knowing that it's millions per microliter. So the red blood cell is in the millions per microliter. It's the most numerous cell that you see. The picture that you're seeing on the right-hand side, that is this picture right here. That is a peripheral blood smear. That's what you do in the office. You can take somebody's blood, put it on a microscopic slide and look at it under a microscope to see exactly what's in their blood. So the first thing to know, it's millions per microliter. Second thing is it's disc shaped. It's not solid. It's not like a donut with a hole in the middle. It's disc shaped. And the reason for that is it allows the red blood cell to bend. When it bends, it can get through the smaller capillaries. That's 7.5 diameter, 7.5 micrometer, and 2 micrometer thick at rim. Those are random numbers, you just have to know. So make sure you memorize those. Now, this red blood cell, the next couple of things is gonna make sense when you realize why it is like this. It originally starts out like any other cell. It has a nucleus, ribosomes, Golgi apparatus, DNA, all of that stuff. Over time, it'll lose everything, so it's essentially a container full of hemoglobin. This has a shelf life of about 120 days. 120 days for this. And after that, it's recycled into pieces, and then by your spleen, if not by your spleen, by your liver. And what will happen is you'll make brand new ones in your bone marrow. So the next definition loses nearly all organelles during development. So there's nothing left. The only thing left in here is hemoglobin. Hemoglobin. Hemoglobin is the transport molecule for oxygen. When you pick up oxygen in your lungs, you have to attach it to something. You attach it to the iron in hemoglobin. And the hemoglobin, think of it like a 4-door sedan. It has 4 seats, right? So the hemoglobin has 4 seats. Hemoglobin is for transport of iron. There's also something called myoglobin, just so you understand the difference. Myoglobin is storage for oxygen in your muscles. Myoglobin is for storage of oxygen in your muscles. Hemoglobin is transport for, oxygen. So hemoglobin is transport for oxygen. Myoglobin is storage for oxygen. They both use iron as the atom for attaching. Now the next 2 points go together. Lack of mitochondria and anaerobic fermentation to produce ATP. Why do you need oxygen?
To breathe.
No.
To stay alive.
Okay.
Support cells.
To get energy.
Hmm?
To get energy.
Correct, ATP, but what do you need specifically oxygen for? That's the general bio, right?
To make H2O.
Cellular respiration. So in cellular respiration, you take a glucose molecule, you break it in half, and then you break it down further in the Krebs cycle, which is in your mitochondria. At the end of the Krebs cycle, you generate a lot of cofactors, FAD, FADH, stuff like that. And then what you do is you create a waterfall of hydrogen. Long story short, you make ATP. So you're able to take one glucose molecule and make something on the order of 36 to 38 ATP molecules with oxygen. Oxygen is the final electron acceptor in the mitochondria. Without oxygen, you can't get rid of those electrons, so it shuts down the electron transport chain, which shuts down the Krebs cycle, so the only thing you can do is glycolysis. Glycolysis is breaking down of the sugar molecule into two pieces. So you originally invest 2 ATP and you get 4 out. So if you invest 2 and get 4 out, how many do you have total? 2, right? You have 2. So 2 net. So when you do glycolysis without mitochondria, you get 2 ATP molecules for every glucose molecule used. If you use oxygen, you can use that same process but go further and make 36 to 38 total ATP molecules. Now everyone sees why oxygen is important. Okay, oxygen is important for you to generate more ATP for the same glucose molecule that you burn in glycolysis. So if your red blood cell is designed to transport oxygen, do you want it to use your oxygen?
No.
No. So hence why it doesn't have a mitochondria, and it can only do anaerobic fermentation, which is to produce ATP.
What is glycolysis again?
Breakdown of sugar.
Breakdown of like sugar. Okay, got it. So,
So that's
basically,
what,
like it's used. That generally, oxygen is used to like generate the breakdown of ATP. Or-
no, oxygen is used to make more ATP.
Oh, sorry, to make more ATP. So it's used like generally for.
To make more ATP
Make
from the
more
same glucose molecule.
Okay.
So, lack of mitochondria and anaerobic fermentation to produce ATP, which means your red blood cell can only make 2 ATP net. Right? So if you're transporting something, you don't want to use it. Does everyone understand that? Yes?
Yeah.
Half of you are like deer in headlights. So
So-
I'm making sure you get it.
So, yeah, I am kind of confused still, because you're saying that the red blood cells, you said that's why they lack mitochondria, because of the whole fact that you want the oxygen to be mainly used for the ATP production, not
In
to.
other cells,
Okay,
not in the red blood cell.
okay,
It'd be kind of pointless if your beer delivery guy drank all the beer before it gets to you.
yeah,
That's the same concept. If you
Okay.
drink beer.
So,
Makes a lot of sense.
so then in red blood cells, why is the oxygen using red blood cells to generate ATP to
No,
generate?
red blood cells are carrying oxygen. They don't utilize it.
Oh, erythrocyte to carry oxygen, right?
Yes. So that's why those two pieces go together. You lack the mitochondria, which means the factory that will use the oxygen, and because you can't use it, you still need to generate ATP. You do glycolysis, which is anaerobic fermentation to make ATP. The other thing it doesn't have is nucleus or DNA. Those get pumped out of the cell, and no protein synthesis or mitosis. So basically, if you do not have any organelles in the cell, this is a product with a limited shelf life, and it'll just start breaking down. When it breaks down too much, all you can do is just get rid of it, break it down, recycle it, and make more red blood cells. So those are those definitions for red blood cells that you do need to know. And that is the way they're gonna ask you.
could we just, I'm sorry, one more time, could you just explain how the hemoglobin is related to the erythrocyte?
So what happens is when you have this red blood cell, when it starts out as the pro-erythroblast, and then it turns into the erythroblast and goes through all the different things, its main job when it has a DNA organelles, ribosomes, is to make hemoglobin. It'll just continue to pump hemoglobin molecules until it fills. But as you run out of space, you start getting rid of the organelles to make even more room. So you're kind of topping it off. So when the red blood cell is mature, all it is, it's basically a bag full of hemoglobin. This thing is designed to carry oxygen, and that's it. So that's all it's doing. So once you reach the maturity stage, you don't need the other organelles anymore, so you get rid of them. And then by the time you get rid of them, the only thing that is in here is just hemoglobin. And hemoglobin is like a sedan, like 4 seats. And those 4 seats allow for cooperative binding. When you do the lecture for blood, you'll learn about cooperative binding. But that's how you carry oxygen from point A to point B. You want this to carry the oxygen from your lungs to your tissues, but you don't want it to utilize it. along the way.
Okay.
So that's why if you don't want it to utilize it, what you don't, what you have to get rid of is the mitochondria. No mitochondria, you can't utilize oxygen,
Thank you.
okay? So those are the definitions that they're gonna ask you for that. They're not gonna add, I mean, if you're lucky, they might point to this and say, what is it? I doubt it. They will point to this and pick one of the definitions, and it'll be your job to pick out the correct definition out of your answer choices. Now, thrombocytes, I don't have a model for that, but thrombocytes are essentially platelets. I'm gonna show you on this plaque in a little bit. So all it is are little bits and pieces of the plasma membrane of the megakaryocyte. Megakaryocyte. So that's a white blood cell in the bone marrow, and what it does is that it breaks off bits and pieces of its plasma membrane or its cell structure, cellular wall, cell membrane, my apologies, cell membrane, and it releases it into your bloodstream. And that's what platelets or thrombocytes are. They're about 2 micrometers in diameter, and they have granules. These granules are essentially packets, and those packets have certain chemicals, molecules, enzymes in them. Now, the number is also important for you to know, 150,000 to 400,000. The exact number, not so much, but knowing that platelets are in the hundreds of thousands, right? Red blood cells are in the millions, your platelets are in the hundreds of thousands. And they last about 5 to 6 days, so a little less than a week. And majority of them are stored in the spleen, so when you need it, your spleen will release. Platelets are used for coagulation. They're used for clotting. When you get a scab on the outside of your body, the scab is a mixture of platelets, fibrin, red blood cells, other debris. That's what a scab is. So when you need to seal a cut, whether it's inside or outside, you have to use platelets. Think of platelets like a brick. When you want to build a wall, if you just layer bricks on top of each other, is that strong? No.
It's not strong.
No. And if you just use a ton of mortar, is not gonna do anything.
No.
So you need bricks plus the mortar, right? So bricks are the platelets, mortar is the fibrin. That's what you need to build a strong seal.
I'm sorry, the red blood cells, the last time,
120 days. Alright. Now, leukocytes. Leukocytes are white blood cells. Sites are cells. Now the number here, again, 4,500 to 11,000. The exact number, not as important, knowing that it's in the tens of thousands. So red blood cells was in the millions, platelets are in the hundreds of thousands, white blood cells are in the tens of thousands at most. You have 3 different granulocytes. Granulocytes. So your granulocytes are called granulocytes because underneath a light microscope, if you were to put these cells, you could see them. They have little granules or little speckle things inside of them, little packets. So that's why you call it a granulocyte. Okay? Granulocyte, because you could see them underneath a light microscope. Now the first one is a neutrophil, which is about 50 to 70% of your white blood cells, which is the largest percentage of all of your white blood cells. The other name for neutrophils is polymorphonuclear leukocyte. Make sure you know that. Polymorphonuclear leukocyte. The granules, even though it says barely visible, are still there, and that is why it's classified as a granulocyte in the cytoplasm. And what these granules are, as you can see on this model, they're little specks that you see. Those specks have tiny little packets of enzymes, and those specks of tiny little enzymes, what they do is they help break down bacteria, viruses, and other things. The other important categorization is the 3 to 5 lobe nucleus. So what that means is this is your entire nucleus for the neutrophil. It's 3 to 5 lobes, meaning it's 3 to 5 chunks separated by thin strands of DNA. So here's 1, here's 2, here's 3. So this has 3 lobes. It can have up to 5 lobes. So what it is the chunk of DNA, small strand, chunk of DNA, small strand, chunk of DNA. That's your 3 to 5 lobes. And it's antibacterial and phagocytic. So phagocytic means it eats up the bacteria and destroys it. So what it does, it engulfs the bacteria and other things inside, and then these little granules will fuse with the bacteria and basically drill little holes to destroy the bacteria. That's how it kills it. So it's antibacterial. When you get a cut and it gets infected and it becomes very tender and it hurts, right? It becomes very tender and it hurts, and then when you squeeze it and that cruddy whitish tan colored thing comes out, that's dead neutrophils. That's pus. Pus is dead neutrophils. So remember antibacterial and remember phagocytic. Right. Next one is the eosinophil. The what? The lobes are just chunks of DNA separated by smaller strands of DNA.
Okay, so that's 4.
That 1, 2, 3.
Oh, that's right.
Yeah. It's 3 to 5 lobes. So this one has 3. You could have up to 5 lobes. All right. Next one is the eosinophil. That's the next most common granulocyte, 1 to 4%. They may or may not ask you that number, but the most common is the neutrophil, followed by the eosinophil. The way you remember this one, 2 classifications, the large red or rosy orange granules, red granules, whatever you want to call the color, but it's red granules. Same thing as this, packets, but these packets have different enzymes that go after different things. And the nucleus is very unique. It's 2 large bilobe nucleus with a small strand of DNA. So that's the difference between the DNA of the neutrophil and the DNA of the eosinophil. It's bilobe, meaning it has 2 large chunks of DNA separated by a small strand, and large red rosy granules, or orange granules. This one again, phagocytosis, which means it has to bring things in. It phagocytoses the antigen antibody complex. So the way, we haven't done this yet, but the way you can think of antigen and antibody. Antigen is what is on your cells that tell your body you're you versus something foreign, right? Like when you go to your home, there's a number on the outside of your home. Everybody on your street knows that's your house number, right? That's your house.
Yeah.
So the antigen is typically on the outside of the cell, okay? So it's like a marker. So what happens is bacteria have certain markers on the outside. What you do is when they enter, you have to tag them. So you tag it with antibodies. Your antibodies are made by your B cells that we'll get to in a little bit. B as in boy. We'll get to those in a second. So when they make the antibody attaches and tags it. Once you tag it, other cells come in and destroy it. In this case, it'll be the eosinophil that comes in to destroy it. So you use the antigen, which is the marker on the bacteria or the pathogen, with the antibody that marks it, and then the part that sticks out is where the other cells attach to destroy what's on the other end. So that phagocytoses the antigen-antibody complex. The other thing you have to know is peroxidase, which is an enzyme that makes hydrogen peroxide, breaks it down, and it's active against parasitic worms. Parasitic worms are any helminthic pathogen that you come across. I'm sure pretty much not all of you, most of you should know what worms are, right? So they're flatworms, roundworms, all kinds of different worms. When you're infected, liverworms, when you're infected, your eosinophil count will go up. And it's also important in allergic responses. So if you take somebody's blood and their eosinophil count is very high, one of two things you should consider are either they're infected with parasites or parasitic worms, or they're having a severe allergic response. One or the other, for the most part. All right, so that's the eosinophil. Now for the basophil, this is the least most frequent cell. This is the rarest, okay? Here, large abundant blue granules versus the red ones. This is blue granules. The nucleus, like it says, it's large bilobed, just like the eosinophil, even though this doesn't look like it. It's bilobed, but it's gonna be very blue granules. So it's very hard to see. Now, increases numbers in chickenpox, sinusitis, diabetes. I don't know why they put it in there, but just know it. It's a random fact that they want you to know. Okay, the important thing of the basophil is that it does two things: anticoagulant, and it's a vasodilator. So what does that mean? Anticoagulant and vasodilator. So first, the neutrophil goes after bacteria. Eosinophil goes after parasitic worms. This does not go after anything. This is a helper cell. What it does, it makes your blood vessels leaky. When your blood vessels are leaky, the vessels, when we went over it, how they're lined by the endothelium, they open up gaps. When the gaps open up, other cells can get out into the tissue. So they allow for other cells to get out of your blood vessels into your tissues. But if you open up gaps in your blood vessels, what is that like? If you open up gaps in your blood vessels, what is that like? Like a cut, right? Your body thinks you cut yourself. So what is it going to try to do?
It's gonna
Clot
be
it.
clotting.
Seal it, right? Clot it, seal it. And that's why it's anticoagulant, because it prevents the seal from occurring, meaning it leaves the gap open for a longer time to allow things to exit. So that is why it's an anticoagulant. And how does it achieve that? Heparin. It makes heparin. So in the granules, you have heparin. Heparin is an anticoagulant. Basically what it does is that it shuts down the clotting cascade, which you do not need to know. For the lab, you don't need to know that, but there's a clotting cascade. If you shut it down, you can't clot. You can't make the fibrin. You can't make the mortar. So even though you have all these platelets floating around, you can't seal it, okay? Now, the other thing it also does, it's a vasodilator. Vasodilator, what it does is it allows the blood vessels to get wider, which means more blood gets to that area, so more things can come out. And so the way you achieve that is with histamine, right? When you have allergies, what do you take? Antihistamine. So what the histamine is doing is allowing the blood vessels to dilate, more things get to that area, more things coming out of the blood vessels, including the liquid portion of the blood. And when that fluid comes out into the tissues, your tissues become leaky because they're oversaturated, and that's why you have a runny nose and watery eyes. When you take an antihistamine, you shut that down. So vasodilation, an anticoagulant, and initiates the inflammatory response. So what the inflammatory response is, whenever you damage an area of your body, you get inflammation. Inflammation is when you get swelling, becomes very tender. The purpose of inflammation is to bring more things to that area to allow for cleanup of your tissues. The problem is when you have too many things coming, they can actually create more damage. That's why you take aspirin or NSAIDs to shut down the inflammation from occurring. So those are the 3 granulocytes. Now, you have monocyte. So now we're at the agranulocytes. So agranulocytes are named agranulocytes because this is a monocyte, this is a lymphocyte. We're gonna get to this in a second. The reason why this is called an agranulocyte is because it does have granules, but you cannot see it under a light microscope. It does have granules, but you do not see it under a light microscope. So this is about 2 to 8%. The nucleus is very prominent. It's a horseshoe-shaped nucleus. It's a horseshoe-shaped nucleus. Now, the reason why it says kidney is you see the cell that's in your picture? Does it look like this? Okay. If you were to take this and look at it on end, would you know it's a horseshoe shape?
No.
No, then it looks like a kidney. So that picture is this cell rotated 90 degrees. Okay, so kidney or horseshoe-shaped nucleus. Now, the third point is very important. It leaves your bloodstream and transforms into macrophages. So it leaves the bloodstream. This monocyte is in your bloodstream. Once it leaves your bloodstream, it becomes a macrophage. A macrophage. The most important thing, it does not go back. It's a one-way trip. From your blood to the tissues, it is one way. Phagocytosis, again, it has to bring things in. Phagocytosis, pathogens and debris. So whenever you have damage to that area, that cell will come out of your bloodstream, get to that area and start bringing everything in, chopping it up and basically eating it to get rid of it. Now, if it comes into your tissues and it can't go back, where is it gonna go?
Does it die?
Hmm?
Does it die
It dies, but where does it go?
into the bloodstream?
It can't go back.
Does it get eaten up by another cell?
Get out of your
Out
body.
of the body?
Hmm?
Out of the body?
I can't hear
It
you.
doesn't get eaten up by
Doesn't get
another
in your
cell.
body.
What? It comes out of your blood, right? Into your tissues. It does its job and it dies. Where's it gonna go?
Disappears?
Disappears.
Like where is
It just
it?
stays there.
Does it?
It just stays there.
It stays there.
Just stays.
Oh.
Stays there. It stays there. It stays there. For those of you with a tattoo, the reason why your tattoo is permanent is because when you deposit ink underneath your epidermis, the macrophages get to that, gobble up the ink because the ink is foreign and it dies and it stays there. That's why your tattoo is permanent. The only way to get rid of it, you have to laser down to the skin layer that has the macrophages, which is usually the dermal layer, to get rid of your tattoo. Just like when you see those commercials with people that smoke or work in a coal mine, their lungs are jet black because all that crap that they're inhaling, the macrophages are breaking it up in their lungs and they die. When they die, they still contain those coal particles, the smoke particles, and they stay there in their lungs, and their lungs are jet black when you do an autopsy.
So macrophages like monocytes like stay in your like body essentially?
Monocytes will stay in your bloodstream. When they leave your bloodstream, they become a macrophage. The macrophages stay in your tissues essentially forever.
But they like so when macrophages die they'll like stain like the tissue essentially? Like
They don't stay in the tissue.
of whatever it's like absorbing.
Yeah, whatever's absorbing. So if it happens to absorb a lot of like coal, if you work in a coal mine, your lungs are gonna be jet black. If you get a shit ton of tattoos all over your body, it'll pick up all that ink and stay Okay? Now, the other thing that it also does is if you watch TV and you get those commercials for DMARD medications, which is basically the skin things, where if you have like autoimmune skin disease and you get dry or like discoloration, they put you on things like Stelara, right? Stelara, or I don't know what, like the newer ones, like 5 or 6 medications. One of the main things that they ask you if you read the fine print is you have to be tested for what disease?
Is it an autoimmune disease?
No, but that's why you're taking the medication. They're asking that you need to be tested for something before that you start on that medication. Does anybody know what it is? TB.
Oh.
So when you have TB, TB is in your lungs. So your body does 2 things. When you have a foreign
Oh.
pathogen in your body,
Oh, then the like isn't like a T lympho.
no, we're not there yet. So when you have TB, right, tuberculosis in your lungs, your body will do 2 things. It'll either try to kill it and get rid of it, and if you can't kill it and get rid of it, what's the next best thing?
It'll adapt to it.
Huh?
It'll try to adapt, right?
Contain. Contain. You wanna contain it, right? You can't kill it, so you gotta limit the movement, you gotta limit the damage. So you have TB in your lungs, but what the macrophages will do is it'll form a wall. Now, if you take Stelara for your skin, the side effect of that medication, which is a TNF alpha blocker, it breaks the wall down. If you break the wall down, what are you releasing?
The TB.
TB. That is why they say you have to be tested for TB before you get to start on the medication, because you don't want to fix your skin problems and end up destroying your lungs. Okay, so that's the other thing they do. They also help form a wall around anything you can't get rid of. If you've ever gotten a splinter, right, and you can't get it out in time, and then you come back like months later and it feels like a bump, that's the macrophages surrounding that splinter. But that little splinter to you is like a gigantic thing for them. It's like you standing next to the Empire State Building, you're gonna chomp at it until you eventually destroy it, which is not gonna happen. So what the macrophages will do is they'll surround that splinter, and that's what you call a granuloma. So the point is, the macrophages, once they come into your tissues, they stay there permanently. Their job is to clean up everything, their job is to break everything down, and if they can't kill it or help kill it, they will wall it off. Okay, and that is why the last line is antigen presentation. So what they can do is once they break up things, once things go inside the macrophage and they break it up, they also show other cells what to look for. So like if you're infected with something, let's say you're infected with staph infection, the macrophages will go in and help kill that bacteria. After the neutrophils destroy it and break it down and release bits and pieces of it, the macrophages will come and clear out everything, and then they will show the bits and pieces of the staph bacteria to the rest of your immune system, so your immune system knows what to look for. So that's antigen presentation. Staphylococcus, streptococcus, they're on your skin. But the main thing is antigen presentation. They will show other cells of your body what the foreign object is, so your other immune cells can recognize it. That's antigen presentation. All right. Now, the next one is lymphocytes. This is about 20 to 40%. So the interesting thing about this cell is that the nucleus is essentially as large as the cell itself. It's a very large, ovoid, round nucleus, very violet or purplish colored. The reason why the nucleus is so large is because it has a lot of DNA relative to the cell size, and the purpose for that DNA is so you can come up with an infinite number, not infinite, you can come up with a very large number of combinations for your antibodies. So for you to come up with different combinations of your antibodies, you need to have that information stored somewhere. This is where you store it, in the nucleus of the lymphocyte. So, if on your practical, because I have done this, this is a lymphocyte. This is either a T as in Tom or B as in boy lymphocyte. If they point to this and say, what is it? And they have B lymphocyte, T lymphocyte, lymphocyte, monocyte, I don't know, neutrophil, something like that, right? Answer is lymphocyte. You cannot tell this is a B or a T lymphocyte unless you do immunological testing, which means not a microscope. You're using assays and other protein-based things to figure out, is it a B lymphocyte or a T lymphocyte? You actually have to test the antigens on the surface and the DNA. So you cannot tell this is a B or a T lymphocyte. The answer you should pick is lymphocyte. That's what you should pick. Now, so the way it works is this lymphocytes, they are made in your bone marrow, in your red bone marrow. They're both made in your red bone marrow. Both the B and the T lymphocytes are made in your red bone marrow. The B lymphocytes will stay in your red bone marrow to mature. And we're gonna talk about what maturation is in a second. But they will stay in your red bone marrow to mature. Whereas the T lymphocytes will go to your thymus, right? Where's your thymus?
It's in the,
Hmm?
it's called.
It's here.
Sternum.
Not here, that's your thyroid. Thymus is here.
Mediastinum.
Yeah,
Mediastinum.
mediastinum,
Mediastinum.
behind the mediastinum. So basically the thymus is located above the heart, between your two lungs, right behind your sternum, right? And on a baby, if you've ever seen an x-ray of a child, they have a large white thing right on their chest. Like the baby's this big and this large white thing is here. That's the thymus. There's nothing wrong with them. That's the thymus. That's normal. So what happens is the T lymphocytes will migrate from your red bone marrow to the thymus to mature. Once they're done maturing, or once you're done with that process, your thymus starts to shrink. So by the time you hit puberty, you're like late puberty, once you finish essentially puberty, your thymus is basically gone. There's a little sliver above your heart, right on the base of the blood vessels, and then it just hangs there. It doesn't do anything. So what is maturation? If you take all the DNA that's in here and you make different combinations of antibodies, I'm gonna show you on the board what an antibody looks like in a second. The purpose is you're trying to generate antibodies, as many different kinds as possible. But what's the problem with that? What's the problem with generating antibodies that are
So
there?
automatic.
If you think of, now I'm gonna rephrase it so it helps you understand. You're gonna train dogs, right? That's gonna be your career. You're gonna train dogs. Inevitably, when you pick up puppies, some puppies will learn to follow your commands and some puppies will learn to bite you. Doesn't matter how much training you give them. Do you wanna give those puppies that are gonna bite you to the new owners?
No.
No. That's maturation for your lymphocytes. You're gonna make antibodies. Inevitably, some of those antibodies will react against your body. Do you want that lymphocyte to go out into the circulation?
No.
No, because if you do, that is autoimmune disease, right? So maturation is you get rid of the T lymphocytes and the B lymphocytes that are making antibodies that are gonna go after you. If you get rid of them, then theoretically, any lymphocyte that's left over should only make things that are not going to attack you. That make sense? That logic? That's maturation in a nutshell for B and T lymphocytes. You're gonna get rid of the B and T lymphocytes, B lymphocytes in your bone marrow, T lymphocytes in your thymus, that are going to be attacking you, because they recognize you, your antigen, as foreign. So if you get rid of those lymphocytes, they will never make it into circulation. If they never make it into circulation, you cannot have autoimmune disease, right? But some people still have autoimmune disease. Why? Because maturation isn't 100%. Some of those do get out into your circulation, and that is why you have autoimmune issues. Make sense? So that's your lymphocytes in a nutshell. Now, the T lymphocytes coordinate immune cell activity. So the way the T lymphocyte works is that it will go up to the cell, physically make contact, and kill it. It has to make physical contact. Kill it. Whereas the B lymphocytes make antibodies, they kind of stay far away and they will release the antibodies, and those antibodies will go and coat the pathogen. And when it coats, other cells will come in and destroy it. Right? So it can coat the pathogen with enough of specific type of antibodies to destroy it by itself, or it can coat enough of the pathogen where other cells will come and destroy it. So B cells kill from far away, the T lymphocyte kills from direct contact. Okay, so that's direct contact. Now, there's a third classification called an NK cell. That's a natural killer cell. And that is a third type of lymphocyte that you have. But the importance of that is your natural killer cell will kill any cell in your body that turns into cancer. That's its job. So any cell that turns into cancer, it'll recognize it and destroy it. That's what a natural killer cell does. So you have three types of lymphocytes, T, B, and the natural killer cell. The B and the T are important because they're part of your adaptive immune system. Adaptive immune system means you can adapt, which means if you are infected, you can adapt, and those particular B and T lymphocytes help you, that help you get over the disease, will now stay on as memory, and they will multiply again when you come across that same infection. So like if you have, I don't know, if you have pneumonia, or not pneumonia, yeah, pneumonia, let's say pneumonia for an example. You have pneumonia, your particular B and T lymphocytes will, like if you have viral pneumonia, they will help you get rid of it, and when they are done, they will resort back and turn into memory cells. So you keep a copy of the cells that helped you got rid of it, and then if you come across that same infection again, they will multiply, but now you don't have to go through the process of trying to figure out which one is it. You already know what it is. So as soon as they multiply, you fight it off.
Rid of it.
Yeah, you fight it off. So when you fight it off, you survive the second time, or you don't even know you have the disease, right? That's the basis for vaccinations. The whole point of vaccinations is giving you something that is not the active. There's very little things that you get that are active. And there's a component in MMR, that's live virus, and your yearly influenza vaccine, if you get the nasal one, are live viruses. Otherwise, you're getting broken bits and pieces of the pathogen, whether it's bacterial or viral capsule or something like that, in the hopes that it'll stimulate your B and T cells to make antibodies. So when you do come across the actual virus or pathogen, you will clear it out, so you do not get sick, right? So that's the purpose of vaccinations. Vaccinations are based because of your lymphocytes. So all of you are going into a medical profession, all of you will come across all kinds of bullshit theories. Vaccines do save lives. Are there side effects? Yes, like anything. You can walk out and get hit by a bus. That's your side effect for going outside. Shit happens, right? So sometimes you have a bad reaction, but it does not mean that's the norm. Those are called outliers. Those are exceptions. The norm is your virus will kill you. Okay, so that is why vaccinations are extremely important, especially with children. When you're born, you get 2 shots in this country and any other developed nation. Do you know what those shots are?
I know one is Hep B.
Right, Hep B. What's the other one?
Vitamin K.
Vitamin K. So you get a Hep B shot, you get a vitamin K shot. Vitamin K is important for clotting. Vitamin K is important for, activates all of your clotting cascade. 902-1070. Those are all the those are all the vitamin K dependent, clotting factors that it activates. Why do you need to give vitamin K? Because where is it made?
In our bodies.
Hmm?
Well, actually, don't vitamin K also come from bacteria
Correct, where?
in E. coli
Correct, where?
in our stomach?
From the mother's.
E. coli's in your ass,
Yeah,
your colon.
that's true.
right? E. coli's in your colon. The baby's GI system, is it sterile or is it seeded?
It's still
Sterile, because they haven't eaten anything, so they don't have E. coli. If they don't have E. coli, they can't make vitamin K. So if they have a microscopic bleed, they will die. So you give them a vitamin K injection to help them clot, or at least kickstart the clotting process in case it's needed. The other one that they get is Hep B. Hep B, if you are a baby and have an immature liver, you have a greater than 90% chance of hepatitis B becoming chronic, which means you have it for the rest of your life. If you're an adult and you end up with Hep B, you have a less than 10% chance that it will be chronic because your liver is mature enough to fight it off. So that is why you give a Hep B vaccination when the babies are born. That is why vaccinations are extremely important, because they do save lives, right? Just like with the big thing now, you guys will come across it, is teaching sex ed to people in high school and early college years, and giving them the Gardasil and other vaccinations for HPV. The vaccine literally saves you from getting cancer. Literally. HPV you get, if you're a female, it'll hit your cervix, your vagina, sometimes the inside of your uterus, but the problems when HPV gets onto your cervix, we'll talk about it in the reproduction lecture, it causes cancer, and you end up with cervical cancer. A male can also end up with penile cancer from HPV. So if you take a vaccine, it'll kill it. What is the vaccine doing? It's showing you bits and pieces of the virus to your body, so when you do come across the actual virus, your lymphocytes will clear it out. That's the whole point of vaccinations, right? What's the BS that everyone hears? What does vaccine cause?
Autism.
Right. That's, when you have retarded people like Jenny McCarthy, and you're like fucking 45, of course you're gonna have a retarded kid, right?
Isn't there also bacteria that causes that?
There's a couple of things you have to identify on the plaque. So first, you see the red dots.
You got erythrocytes?
These are the red blood cells, the erythrocytes. Then you see the clear cells with the 3 to 5 lobes here, 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 and here. That's the neutrophils. Correct. It is a leukocyte, but neutrophils. Then the bilobe nucleus with the red, eosinophils. There's 3 of them here, here, even though it doesn't look like that, and here. Then you have one basophil right there. Okay, one basophil. Now, platelets are these little blue things. Okay, those are platelets. What is the only other cell that's blue?
The lymphocyte.
Correct, they're sky blue, but not this color.
Wait, what?
The granular.
The
Omega
lymphocyte.
carrier cell. You see the color for the platelets, they're blue? This is the only cell with a blue cell membrane.
What is, which one is that?
Omega carrier site.
Okay.
Mega carrier site.
Are these the lymphocytes?
This is a mega carrier site.
Oh, the blue part, right?
Yes. Mega carrier site.
Oh, okay.
This and this are lymphocytes.
Ah,
Okay, this is a mega carrier site. Do
Mega
not make that mistake. The other thing is this and this and this is what?
this is the
Monocyte.
Monocyte.
Do not pick macrophage. Macrophage will be one of the answer choices, and it will be wrong, because where is this?
In the
your
blood.
In the bloodstream. So monocyte, 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 megakaryocyte. If you forget, the platelets are blue, has a blue plasma membrane. Your 2 lymphocytes, basophil, eosinophil, neutrophils, and then the red blood cells. That's the plaque.
No monocytes?
Hmm?
No monocytes?
Yeah, right here. Here, here.
One looks like a horseshoe, right?
Yeah, I mean, they're not gonna be like exactly